So hello everyone and welcome to our presentation for this morning. I'm just going to put up again the deck that we had there a moment ago. My name is Dawn Van Dam and I am the Executive Director of 1 Million Solutions in Health. So I would like to welcome everyone today to this presentation. I'm very excited to have you all here and to be sharing with you some fabulous science. So as I said, this is the NIH licensing opportunity. We're going to be looking at a novel diagnostic imaging platform to monitor the progression and treatment of metastatic cancers. So welcome to this session, and I'm just going to jump right in. There we go. So today's agenda is I'll do some welcome and introductions. Our presenter today is Dr. Stephen Stern, which I'll tell you a bit more about him in a few minutes. We're going to also ask you to be thinking very carefully about this science and help us evaluate it and help us consider how you can work with us to bring it to market. There'll be some closing remarks and ask you to complete a survey for us. So our vision as 1 Million Solutions in Health is to accelerate the discovery, development, and delivery of innovative cures, treatments, and preventative measures for patients around the world. So fundamentally, we try to bring new science to you all along the way. We have, as part of that, our Hidden Gems program, where we basically help people get the edge in the marketplace for science and help bring things to market even faster. We get our science from all different areas and work with all different parts of the industry, whether it be scientists or organizations like the NIH, or academia, entrepreneurs, and industry. We also have a technology evaluation consortium where we bring together pharmaceutical and biotech companies with technology vendors or scientists to help them again accelerate that pathway to market. This is just a sampling of some of the people that we've worked with over the years from the industry side. There you go. Hopefully you recognize some of those people or some of the companies or some of the names. Okay, so through our extensive network, we find these new scientific solutions. We gather input to optimize the technology. We share them with our 1 Million Solutions in Health community as a charitable nonprofit. And then we also have a program where we work with you to help commercialize them and optimize them for the marketplace. So this program today is part of our Signature Square program, where you're going to help us evaluate things and ask questions and understand how you could either license it or use the CRADA program, which I'll get into in a moment, with the NIH. So our Signature Square is the evaluation tool that we have, and here's the program that we work with the NIH. So at the NIH, of course, we all, as people who are taxpayers in the United States, or people who can benefit from the work coming out of the NIH, we want more licensing agreements to occur to help bring their great science to the marketplace. There's also the opportunity earlier on in the process to work with the NIH in the process called a CRADA, or Cooperative Research and Development Agreement, to help move along the science that needs some more work to help bring it to market and finalize it and get it ready for launch. And of course, today what we're looking for is feedback and direction for their future path and input into their roadmap, and for partnerships on the first 2 bullet points there. So with no further ado, let me give you a little bit of background about our presentation today. As I said, this is a novel diagnostic imaging platform to monitor the progression and treatment of metastatic cancers. This novel technology is a polymeric delivery platform that targets the lymphatic system through scavenger receptor A1-mediated transcytosis. This receptor is highly expressed in macrophages, monocytes, mast cells, dendritic cells from the myeloid lineages, and endothelial cells. The platform can deliver various diagnostic imaging compound cargo to the lymphatic system and myeloid antigen presenting cell, APC, subpopulations that can be used to track the progression of various disease states or the progress of an associated treatment therapy. This lymphatic system targeting polymeric delivery platform is particularly well suited for diagnostic activities associated with monitoring the progression and treatment of metastatic cancers that use the lymphatic system to spread to other parts of the body. So let me do a quick introduction here of our speaker for today. Dr. Stephen Stern is laboratory co-director, director of R&D, and head of the pharmacology and toxicology section of the nanotechnology characterization laboratory. His areas of expertise include biochemical toxicology of the liver and kidney, analytical methodology, DMPK, and nanomedicine formulation. Prior to joining the NCL, Dr. Stern was a postdoctorate fellow at the University of North Carolina, Chapel Hill. He received his BS degree in biochemistry from the University of Rochester and his PhD in toxicology from the University of Connecticut at Storrs. Dr. Stern is a diplomate of the American Board of Toxicology. So I'm going to switch at this point to him. Let him bring you the presentation for today and switch it from that perspective. I will ask you though, if you could put your questions into the chat box at the bottom of the screen. So if you hover over the bottom of the screen, you'll find the chat box. And if you could put your questions in there after his presentation, we will then move to a Q&A, which I will moderate. So I need your questions down below so that I can ask him your questions. So we're seeing a Word document on your screen, Dr. Stern, rather than your PowerPoint.
Okay, yep. Just trying to get this back on the right screen.
Now we see your, just your computer with hundreds of documents on it.
Yeah. Just a second. I think it should pop back up. It's a bit slow.
Touching it now.
Here we go.
Yeah, great.
And let me just put the full screen mode on. I was trying to move my screen over unsuccessfully, unfortunately. Sorry if you heard me clear my throat while Dawn was talking. And Dawn, I appreciate the introduction. I do have a bit of an allergy, so hopefully my voice stays with us today. But again, I thank Dawn for the introduction. So my work in nanotechnology spans a course of about 15 years, working on drug delivery, diagnostics. My real forte is novel formulation and pharmacokinetics, as well as toxicology aspects of nanomedicine. And so it's pretty broad. It would be broadly placed within pharmacology and toxicology of nanomed. So today, I'm going to be talking about this novel polymeric prodrug system that can be used both for therapeutic applications and imaging applications, but today we're going to focus on imaging. The platform uses a scavenger receptor A1 targeting to gain entry into the lymphatic system and lymph nodes in particular, and within those structures targets myeloid cells by the same scavenger receptor A1 uptake mechanism. So to give you some background on this technology, the lymphatic system, as you probably know, is responsible for mounting an immune response against foreign pathogens. Additionally, it's a conduit or circuit by which immune cells move about the body to sites of infection and injury, and do so bypassing the blood, the systemic circulation in the process. Unfortunately, cancer cells have also been known to utilize the lymphatic system for metastasizing or spreading throughout the body. And so it's very common, especially with certain cancers like breast, melanoma, head, neck, urogenital, that these cancers will spread into the lymphatic system, and this is how they gain a foothold through the rest of the body. So very often in surgical removal of a primary tumor, be it a breast cancer or melanoma, they'll then seek out the sentinel lymph node, which is the most proximal lymph node, to determine by biopsy if it is cancerous, that is to say, if the cancer is metastasized to that sentinel lymph node. And if it has, they would then look at additional lymph nodes. So imaging of sentinel lymph node is a very common biopsy and surgical procedure for identifying cancer spread and for allowing for surgical removal of a cancerous lymph node. Now, lymphatic imaging in general, in addition to sentinel lymph node imaging, can be used to monitor cancer disease progression, to look at lymphadenopathy, which is the enlargement of a tumor, and to follow treatment courses of the disease and stage disease as well. So there's a lot of interest, especially for cancer, in the application of diagnostic lymphatic system imaging. So how do we go about monitoring the lymph nodes for surgical removal for identification of sentinel lymph nodes with current technology? So the majority of the imaging drugs used to identify sentinel lymph node include radiotracers, fluorescent dyes, and superparamagnetic iron oxide nanoparticles. So these are nanoscale materials, these SPEOs. Other sulfur colloids are used as well, which could be considered nano. And the reason for the nano aspect is that these tend to stay within the lymphatic space and they track within the lymphatic system. And so these colloids are often used and can passively accumulate within lymph nodes. These are then once introduced either by intratumoral or peritumoral administration, such as with breast cancer and melanoma, as I mentioned. This movement to the lymph node and identification of the lymph node is conducted by gamma probes, fluorescent cameras, and handheld magnetometers during the course of surgery to allow for biopsy as well as removal in the case of a cancerous lymph node. So the problem with this current system of using these non-targeted drugs is that these drugs simply diffuse through the lymphatic system upon administration via these intratumoral or peritumoral administration routes, eventually arriving at the lymph node where they are then often taken up by immune cells. This passive distribution leads to a high background signal and not an ideal contrast between the lymph node and surrounding tissue. So again, this is not an active targeted process. By comparison, active lymphatic targeting results in much greater lymph node contrast, as we'll show today, and improved removal and surgical outcomes. So there is a class of agents used for sentinel lymph node identification that are billed as lymph node targeting. And I wanted to go into these specifically as they're the only class of materials currently used that at least purport to target lymph node. And they do so through a mannose receptor targeting. Now, mannose receptor is found on macrophage within lymph node, okay? And so these agents have to diffuse to and into lymph node and then interact with these macrophage and be taken up by them in order to accumulate within the tissue. There are problems with that, okay? Mannose receptor targeting agents, again, will lead to higher background because it's not an active uptake into the lymph node proper. It's only a diffusion into that tumor with then active uptake into immune cell populations within that lymph node, such as myeloid cells, such as macrophage. Targeting agents currently on the market, Lymphoseek, Magatrace, they all use a dextran polymer ligand in order to interact with that mannose receptor. And this dextran chemistry has been associated with anaphylaxis and other immunotoxicities. And for this reason, both of these products have FDA warnings of anaphylaxis potential. And so there's obvious liabilities with the current marketed quote unquote lymph node targeted agents. Additionally, these current marketed agents use magnetic resonance in the case of Magtrace and radio tracer technology using technetium and SPECT imaging in the case of Lymphoseek. And this adds to the cost and safety concerns with running these probes. Obviously, radio tracer, you have to deal with the source for making the radio tracer, which has to be close or within proximity to the hospital. You also need to dispose of these materials, and this adds to safety concerns. In both the MR and radiotracer case, you're also dealing with expensive diagnostic equipment. Ideally, use of a fluorescent probe, a fluorescent dye, is much more cost effective and often are still in use. Blue dye and and green dyes such as indocyanin green are often in use for doing the same purpose as these targeted agents, but they can use visual or fluorescent microscopy to identify the lymph nodes. And this makes for a more cost-effective means of identifying and removal. So the invention that we're going to be talking about today is polylysine succinylated delivery platform. Now this platform is based on a polylysine backbone, so that's a polyamino acid made up of lysines, upon which the pendant amino groups are succinylated with the succinyl group here, which then changes this amine functionality eventually to a carboxylic acid functionality. And we found that this succinylation, specifically the succinyl group itself, was able to interact with scavenger receptor A1. And this is well known, actually, to be a potential substrate for scavenger receptor A1 being polyanionic polymers. Okay, so this was a new novel finding, but it fit with the other substrates that have been identified in the past. The free carboxylic acid groups on these molecules, these pendant carboxylic acids, then allow for attachment of therapeutics through esterification or imaging probes through stable amide bonding. And I'll go into that in the next few slides. But the important take home is that this platform can be loaded with drug through conjugation. The drug is released in a controlled fashion for therapeutics, and it is not released, it's very stable for imaging. And these agents interact with scavenger receptor A1, and this allows delivery into the lymphatic system. As far as what agents can be attached, we've attached immunomodulatory and antiviral cargo, such as small molecules, therapeutic peptides, vaccines. We've also attached imaging dyes and shown that there is uptake of these prodrug systems in the case of therapeutics or macromolecular imaging dyes to lymphatic and lymph nodes through the scavenger receptor A1 mediated transcytosis, resulting in uptake eventually into myeloid cells of sentinel lymph nodes upon intradermal administration or global systemic lymph nodes upon IV administration. There's also been shown uptake into tumor-associated macrophages associated with cancer. So the proof of principle studies that were the important studies initially in these findings were to show that macrophage, specifically murine raw 264.7 cells, in which scavenger receptor A1 was knocked out, did not take up the polymer, even after 24 hours of incubation. However, wild cells that contained scavenger receptor A1 expressed this receptor were able to take up the polymer rapidly and in all cells that were exposed. And so we were able to show very specific uptake dependent upon this one receptor, being scavenger receptor A1. In mice, upon administration of a dye-conjugated PLS construct, we were able to show rapid distribution to the lymphatic system in large and also lymph node distribution. And this is going to be the majority of the data that we focus on today as far as application of this technology for use in imaging of the lymphatic system and specifically sentinel lymph node identification. And that's the main summary of the technology or the take home of the invention. So the way that this invention works, the dye conjugated polymer can be delivered IV or intradermal. Upon IV administration, the free succinyl groups interact with scavenger receptor A1 that's found on both the vascular endothelium, and this is important, it's also found on the lymphatic endothelium. So once interacting with the endothelium of the vasculature, it undergoes transcytosis. This is a receptor-mediated transcytosis, which allows for uptake into the lymph, and then through lymphatic scavenger receptor transcytosis, uptake into the lymph node itself, and then eventual uptake into the myeloid cells, also through the SRA1 uptake pathway. And so this is a rapid mechanism by which this dye in the blood is incorporated into the lymphatics and into myeloid cells. Now we can administer this dye polymer by IV, sub-Q, or intradermal administration, and each has its own benefits. We'll mention that. This active uptake into lymphatics and lymph node, of course, is a SRA1 mediated transcytosis process and occurs through both the vascular and lymphatic endothelium. This is what makes it a truly lymphatic targeted technology. We're not using passive diffusion to eventually get into lymph, such as if you administered a SPIO, a superparamagnetic iron oxide, into the blood. It takes on average 24 hours to light up the lymph node. This technology will light up lymph node as early as a half hour after administration. At least that's the earliest time point that we looked. It could be even earlier than that. It also has superior chemical versatility. So we can attach any amine through amide chemistry and amidation to this succinyl group. And for drugs, any group with the hydroxyl, free primary hydroxyl, we can attach through esterification. So there's a lot of versatility as what can be attached, because even if the dye itself does not contain an amine, very often there are versions of the dye that have amine functionality. So that's something to keep in mind. So for drug esterification, for making a prodrug, we use click chemistry, that's carbodiamide chemistry, to attach the drug to the carboxylic acid group through this ester. And this ester is hydrolyzable both in a pH-mediated and esterase-mediated way, both enzymatic and non-enzymatic. And we have found the release characteristics have controlled release properties. It has to do with the number of carbons and the functional groups within this linker that allow for an ideal release half-life of between 10 and 40 hours consistently for over 20 different drugs that we've attached to this molecule. We also have very high drug loading, and the efficiency of drug loading is also very high. So drug isn't wasted in the process of loading the polymer. This can be used then for drug therapy of metastatic cancer in lymph nodes, treatment of lymphatic infections such as HIV, or immunomodulation such as in cancer. This linking of a free drug to the polymer decreases systemic drug concentrations while increasing drug concentrations at the target tissue, being the lymphatic tissue. So this results in reduced hepatic clearance and reduced side effects. Now, what we're focusing on today, of course, is the imaging aspect, in which, while in therapeutic or prodrug synthesis, we'd like a controlled releasing API. In the case of a imaging agent, we want a stable construct. We want the dye to stay with the polymer that is then targeting the dye to the lymphatics. And so in this case, we're using amide coupling, which is also simple carbodiamide click chemistry with very high yields. So we get very high drug loading. Again, drug efficiency is very high. There's very little wasting of drug. Most drug is incorporated into the polymer, if not 100% incorporation. And the stable amide is not easily hydrolyzable, and so the dye stays with the backbone that is responsible for the drug delivery. This is active lymphatic targeting. It's rapid. This can be used to monitor lymphatic disease progression, to see where the cancer has spread into the lymphatic system, to see how the cancer is affected by treatment, therapeutic effectiveness. And of course, A very simple application is its use as a identifier of the sentinel lymph node. So here is an example of some of the in vivo data that we've generated with several different dyes using a 62 kilodalton PLS dye construct. So this is the size of the polylysine succinylated polymer that we use in these studies, and size is important, and I'll go into why. So here, upon IV administration, okay, of the 62 kilodalton PLS dye polymer, we see rapid accumulation to within the liver and the spleen, and this occurs within a 30-minute time span. We see this uptake into these tissues, okay, in vivo. What we're seeing here in this ex vivo imaging is 24 hours following the IV administration, but at 24 hours, you can see there is a very clear high fluorescent contrast between these inguinal lymph nodes, which we have taken out with tissue to show that there is no background. I mean, you can observe just with your naked eye here how easily it would be to identify during surgery for removal if these were the sentinel lymph nodes. But of course, this was administered IV, so this went to all of the lymphatic tissue and lymph nodes in the body and did so rapidly. So this would be an indication in which you were trying to image all lymph nodes to identify potential of unknown lymphatic metastasis of a cancer. And here we're using this cyanine 7.5 near infrared dye, which again has this amine functionality for incorporation into the polymer. Next, we looked at a different dye, but same size construct, 62 kilodalton PLS. Here we're looking at Alexafluor 488, and we're using immunohistochemistry, that's antibodies to the dye, to have high resolution imaging of where the dye is going in tissue. And again, this has that amine functionality to allow the stable amide coupling to the polymer. Upon IV administration, we see uptake generally to all lymph nodes in the body, mesenteric, popliteal, axillary, inguinal, in equivalent amounts. And this is 24 hours after injection. If we, however, administer this construct by intradermal foot pad, it tracks to the most proximal lymph node, being the popliteal lymph node, and lights that lymph node up. And this occurs as early, again, as 30 minutes. At least that's the earliest time point we've looked. Here we're looking at 24 hours, and you see even at 24 hours, with a very high injection concentration, we see little spillover into the other organs by comparison to this organ. So obviously, if you were to administer a small amount, you would get uptake into the popliteal lymph node without the dye venturing any further into the lymphatic system and other lymph nodes. The potential advantage of active targeting for sentinel lymph node imaging is that you decrease the drug requirement. As I said, you can give very little drug because it's so targeted to the lymph node and then to the myeloid cell population within the lymph node. This increases contrast, as you're seeing here, as you saw on the previous slide, and this allows for easy surgical resection with improved surgical outcomes. Now, lastly, I'd like to talk about the size dependency of intravenous PLS lymphatic targeting and imaging, and how this also relates to renal clearance. So the nice thing about these polymeric constructs is that they're tunable. As you change the size, you can change the distribution characteristics. Again, as I said before, IV administration is not for sentinel lymph node identification, because sentinel lymph node requires tracking of lymphatics from the primary tumor or site of injection to the first lymph node within that lymphatic tract. But if you want to look at global lymphatic lymph nodes, all lymph nodes in the body, all lymphatics, then you want to use an IV administration, which is taken up into the lymphatics, identical to how it occurs in the intradermal administration, but in this case, going through the vasculature into the lymph, and as I described in the cartoon, into the lymph node through the lymphatic endothelium itself. And what we found is that upon IV administration, with decreasing size, there's an increase in lymph node identification or lymph node signal in comparison to liver. So here we showed a ratio of lymph node fluorescent signal to liver, and as we decrease size, we dramatically increase the lymph node uptake, decrease liver uptake, and we increase renal clearance. And the reason for this is that the 10 kilodalton PLS construct is below the size of glomerular filtration, which is 5 nanometers. And so here with the 10K construct, we see a very rapid clearance in the urine in comparison to the larger constructs, which still clear, but have to be broken down by that polymer backbone before it's of the size, around 10K, to which it could clear as well. So that's another nice thing, is that even the larger constructs, as might be used to track within the lymphatic system for sentinel lymph node imaging, like the large 62K structure, that eventually is going to be broken down and would be of size to clear renally. And we see this clearance, as you can see here, even for the larger constructs, even though initially they would not clear. So this shows the rapid degradation of the polymer. So the potential advantages, again, of IV active targeting is you have systemic lymphatic imaging. This occurs rapidly. This is an active targeted process. You don't have to wait, such as with an iron oxide system, for it to percolate into the lymphatics and eventually light up the lymph node. Here, it's going through the vasculature, through the lymphatic wall or endothelium, if you will, and into the lymph node. And this is a rapid process, so it decreases the drug requirement. Whenever you have active targeting, you don't need as much drug to get the same contrast. And at the same time, because you're loading more drug into the actively targeted tissue, but also you decrease your background by using less drug. So it's a win-win for visualization of the targeted tissue. So currently, this is in the national stage. National stage is pending. As far as claim coverage, the claims include method of delivering therapeutics and diagnostics using the PLS delivery platform. Just a second, I'll take a glass of water. Okay, and compositions comprising the PLS platform. It also includes specific compound compositions. These are for immunomodulation, in the case of PI-103, which is a PI3K mTOR inhibitor, as well as emtricitabine and lamivudine antiviral drugs used to treat HIV. Those are included as specific compound compositions. And as far as potential commercial advantages, just to bring this home, again, it's all about active transport. If you can get active transport into the lymphatic system, into the lymph node, and then within cells of the lymph node, such as myeloid cells, this is a much better system than the current targeted systems on the market, which only targeted cells within the lymph node, not the pathway to get to lymph node, as well as cells in the lymph node. And I hope I brought that point home. The platform is very simplistic in its chemistry. It's biodegradable. It's compatible with the majority of imaging agents that we've tried so far to load, and we've tried several. There's increased contrast because of that active targeting, and this can lead to improved patient outcomes, either through identifying therapeutic efficacy or improving surgical removal of lymph nodes. The requirement of drug required for lighting up the lymph node is decreased. And this in and of itself improves the safety profile, obviously. But also by decreasing systemic concentrations, as targeted systems do, we're reducing the drug toxicity and the side effects of the drug at the same time as actively targeting the drug. So those are the major potential commercial advantages. As far as what we're looking to do with this technology at this time, we're still in the early preclinical stage. We've conducted efficacy studies in cancer models for therapeutics. We've conducted these imaging studies of the lymphatics, and we've conducted initial safety studies. What we're looking to do now is have a group come in, license, and assist with further development, ultimately leading to a clinical trial. Or if a company would like to take on development themselves, that would be great as well. As you could see from the claims, they're fairly wide open. A drug could be incorporated into the platform that a company is currently working on. This could be for imaging, of course, for therapy, immunomodulation, etc. So we're open to any types of collaboration. So that's the end of my presentation. For any interest in collaboration and licensing, I would point you to Lauren. And I would be happy to take any questions that you may have now. Thank you. Thanks for the interest.
Awesome. Thank you so much, Dr. Stern. We'll just leave that slide up there for now. That's great. Actually, you could just leave it. That way everybody can copy it down as we go forward. So we have a couple of questions that have come through. So I'm going to start with those, but I'll just encourage everyone who's watching to put in any more questions into the chat box. You just go to the very bottom of your screen and highlight that. Do you want to just put that back into full screen maybe?
Okay, just a second. Okay, there you go.
Yeah, so that way people can get all the proper spelling of everything if they want to contact you or Lauren. So yes, Lauren is the technology transfer manager, as you can see there at the NIH, and she'll help walk you through the process of, you know, the options in regards to a license or a collaboration, CRADA. And Dr. Stern, of course, can tell you more about the science. But let's get over to the questions now. The first question is, what level of magnitude are you referring to when stating, quote unquote, less drug is needed?
So what we've seen in targeted systems, using this targeted system in comparison to a fluorescent dye that is known to diffuse into the lymphatics, the comparison in fluorescence of, say, the lymph node was at least 50-fold. It was dramatically higher than just the diffusible drug. So if that corresponded to the amount of drug introduced, this was not, well, this was an intravenous study, but in that case, certainly if everything had been linear with regard to signal and dose, we would expect we could dose 50 times less of the imaging agent and see equivalent signal to that of the free drug. So we saw, you know, at least 5-fold, you know, if not even an order of you know, 2 orders of magnitude potentially.
Okay, awesome. And again, if anyone has any follow-up questions to any of the answers, please put those into the chat box or the Q&A as well. So here's another question for you. What imaging modalities would this platform accommodate?
So I talked about the chemistry being very versatile, right? So basically any amine functionality can be conjugated to that succinyl ester through the simple, you know, click chemistry anidation. And so if you look at the potential introduction of probes using that simple specification, we've obviously incorporated several fluorescent dyes. I know we haven't yet, but we could certainly incorporate indocyanine green, which is currently in use as a sentinel lymph node identification dye. And this would dramatically improve that dye, I'm sure. SPECT and MRI probes could be incorporated, as we could incorporate chelating agents. We have done that in one case. So you could incorporate technetium for SPECT. You could incorporate gadolinium for MRI. And of course, you can incorporate F18 for PET imaging. So I think the it's really just an open question as to what the benefits are of each of these and how you want to use the probe. But certainly any of these technologies I mentioned could be used.
Okay, terrific. Thank you for that. Another question here is, how do you synthesize these polymers? What's the average degree of polymerization?
So we actually purchased the polymers at this time. We purchase the polymers, but then we succinylate them ourselves. So we're buying polylysine very often and then succinolating it. And the succinylation is 100%. It's a very robust process using succinic anhydride. But maybe if you were getting at to the drug loading. So drug loading is typically between 10 and 20% by weight of the molecule. Okay, so that's pretty high loading for a conjugated system such as this. Remember, we have to still have 3 succinyl groups that allow for the targeting. Obviously, if we were to incorporate drug at every succinylation site, first of all, if you were dealing with a hydrophobic molecule, you would then make an insoluble structure. But also you need those free succinyl groups to interact with the scavenger receptor to allow for the active transport process. But that being said, we see loading of 10 to 20%, and loading efficiency is usually above 80%. So 80% of the drug we incorporate into the molecule, that incorporation is 10 to 20% of weight.
Okay, terrific, awesome. Another question here is, have you done studies examining the protein corona effect and whether that changes the targeting ability in vivo?
So we have not looked at protein binding of this structure. Certainly we've worked in very low protein systems in vitro, in which we were able to examine this interaction with the scavenger receptor. Because of the anionic nature of this polymer, it's quite anionic, of course, with all those carboxylic acid groups. I suspect that binding to protein would be very low. And again, if you think about the fact that polyanions are typically substrates for scavenger receptor, and there's also very early work succinylating proteins that showed interaction with this scavenger receptor, we really believe that uptake is dictated by a free succinyl group interacting with that scavenger receptor. So if there is protein binding, it must be fairly minor as to not obscure that targeting.
Okay, thank you for that. And again, if there's any follow-on questions, please add those into the chat box. Is there any safety data available?
So there is safety data available. Now, these weren't pure toxicology studies. What we did was tack on hematology, clinical chemistry, and hematology to imaging and anti-cancer efficacy studies. In those studies, we saw no evidence whatsoever, no indication of toxicity, no increase in liver enzymes, no kidney markers, no histological changes, organ weights. For the platform. In cases where drugs were incorporated, we saw much lower toxicity for the drug incorporated than the same dose of the free drug. So the platform decreased drug toxicity, as we would expect, though there was some evidence of toxicity related to release of the drug, of course.
Okay, thank you for that. What is the half-life of the drug with this drug delivery system?
So the uptake into tissue is quite rapid. Okay, the uptake is within 30 minutes. You start to see at least an increase in lymphatic distribution, but the half-life itself is somewhere around 3 hours. So within 3 hours, and it's dependent on the size of the PLS. Okay, so I'm talking about what we've worked with the most, which is the 62 kilodalton. 62 kilodalton has a half-life of around 3 hours. So within, within 2 or 4 half-lives or 6 half-lives, if you're a physicist, it would be completely removed from the system. But, it's a fairly quick half-life, and we believe that's really being the half-life of it being cleared renally. It's being chewed up, also incorporated into tissue, but the tissue incorporation occurs very, very rapidly. So, yeah, I think the tissue loads up very quickly, and then it's slowly degraded and cleared by the kidney. If we're looking at the small 10K construct, that construct immediately goes to lymphatics and is immediately cleared by the kidneys. So that's a very rapid release and decrease in background. And that's really an ideal size, I believe, for systemic administration, for looking at global lymphatics, because what you want is, obviously, you want the dye to go to where it's supposed to go, and then the remainder to be cleared so that you don't get bioaccumulation of the drug, decreases your safety concerns, and also, importantly, increases your contrast to the targeted tissue versus the blood concentration or even interstitial concentrations.
So if there was a longer surgical procedure, you would just adjust things how?
So the half-life in the systemic circulation is what we were discussing. That's what I was discussing. So, so the fact that it goes to the site of action, being the lymph node, very rapidly, and then clears rapidly as well from the systemic circulation, the circulation time really isn't a concern because ideally you'd like it to be cleared immediately after it gets to where it's supposed to be going. Within the lymph node, as you saw from the the studies that I showed, and I can go back, here, this immunohistochemistry, we're looking at 24 hours out. Okay, so at 24 hours, we're still seeing it sitting in the lymph node. Same thing here, this was for intradermal, and this was for systemic. We're still seeing it there at 24 hours. Now, these are within myeloid cell populations, and they should break down and clear, and that's certainly what's going on in the tissue, but we haven't looked at a tissue half-life. We can only say that we see good signal as early as 30 minutes, and that signal is still good at 24 hours, even though the drug's no longer in the systemic circulation, or as you can see in the surrounding tissue. It's really just where it was supposed to go, being the myeloid cell populations of the lymph node.
Awesome. Great. Thank you. So before we started today, this morning, we were having an offline conversation about the current methodologies, and in particular, the issue around anaphylaxis. Do you want to discuss that a little bit at this point?
Yeah, sure. So anaphylaxis, I guess this is in regard to the dextran targeted agents, or just in general?
Just in general, why this would be a better alternative, and there's an issue with other alternatives that could perhaps be addressed here.
So anaphylaxis, as I'm sure everyone on the call, knows to some degree, is an issue for any infusion, right? So any infusion potentially can cause anaphylaxis. It tends to be, it's an idiosyncratic effect. For agents that are more likely to cause anaphylaxis, such as doxorubicin liposome, you might pre-medicate with steroids, dexamethasone, antihistamines, diphenhydramine, to dampen any response. But that being said, there are certainly agents that are more prone to anaphylaxis. They just have a historic history, of anaphylaxis. But in particular, as we're discussing here for imaging agents, dextran-coated superparamagnetic iron oxide nanoparticles are notorious for inducing idiosyncratic anaphylaxis. And this anaphylaxis, it's idiosyncratic. It's not seen in every individual, but it's in a high enough proportion of individuals that it's known to the medical community, and it is a concern. In the case of Doxil, it happens at such a high rate that you do actually pre-medicate patients, taking Doxil to prevent that potential reaction. But obviously, for this type of study, this diagnostic use of a dextran-coated material, now it could be the Lymphoseek, which is a dextran polymer with technetium attached to it, or it could be the Mega scan that's in iron oxide with a dextran coat. In either case, the dextran coating or the dextran polymer, it has been shown to be the bad actor in inducing anaphylaxis. So if we can remove that chemistry and still achieve targeting, and achieve targeting in a better fashion than those agents, that would be ideal. So obviously our polymer is free of any dextran moiety. And additionally, the chemistry of our structure, it's not a sugar structure. It's a polyamino acid that's been succinylated. We don't have any evidence at this point of anaphylaxis, but of course we have no clinical experience with the drug, and animal models are very poor. But that being said, we don't expect it to be a bad actor based on what we know of agents that are bad actors that have gone into the clinic. So that would be an improvement in safety.
Great, awesome. Thank you for that. Another question here. We've got some really great questions today. Is there any limitation for conjugation of the imaging dye or the drug to this delivery platform?
So some limitations or some things that you may have to work around is if you have multiple functional groups that could undergo the carbodiamide attachment chemistry. Now, carbodiamide chemistry can be geared towards esterification or amidation primarily. And so you can use specific reaction protocols to either make an ester if you have a free hydroxyl group, or make an amide if you have a free amine. So that's not so much a problem if you have a molecule that has both functionalities, okay? Hydroxy and amine, you can force the chemistry to one conjugate or the other. The problem comes in is if you have multiple hydroxy, primary hydroxyl groups. If you have multiple amines, potentially you can form gels when you make that. And so if that's the case, you may have to block one of the amines or one of the hydroxy groups. We've actually had to do this recently, but it was very successful. You just had to put a blocking conjugate onto one of the functionalities to keep it from cross-linking in the reaction. That would be the primary concern. But other than that, we have been amazed, okay, at the types of molecules we've been able to attach, from very hydrophilic molecules to extraordinarily hydrophobic. It's really been a wonderful platform to work with for that reason. I can tell you now, we have attached peptides, we have attached chelating agents, we have attached many, many dyes, we have attached up to almost 10, I think, small molecules. We've attached nucleotides, okay, oligonucleotide. So it's really, really a diverse set of chemistries, with those functional requirements that we've been able to attach. And in some cases, you do have to modify your API. You have to put on some type of functionality to get it to work, but that's not often very difficult to do.
Awesome. Thank you for that. I'm going to give you two questions because they're very related.
Okay.
So have you tried any other drugs other than HIV drugs in your in vivo system? And the other question is, in addition to imaging of lymphatic tissues in metastatic cancer, what other tissues, disease states might be imaged with this platform?
So the answer is yes. We have tried, and I can't go into the specific drug, okay, but because it's some of those those actual new compositions are undergoing patenting, or in some cases they just haven't been presented in any format. But I can say we've looked at several immunomodulatory drugs for cancer. We've looked at adjuvants for vaccination, because obviously the system goes to not only myeloid cells in lymph node, but also to antigen presenting cells like dendritic cells in the skin. Okay, so it's a very good means of getting an adjuvant to an APC. Several different cancer drugs, cytotoxic agents. We currently have an ongoing study that's going on right now, preclinical study for a melanoma drug, and I can't go into the specific of the drug, but this drug has been around for 20, 30 years, okay? And no one's made a formulation that achieved several different criteria, being long exposure, decreased neurotoxicity, et cetera, et cetera. And we think we finally have a formulation of this very old natural product that's based on the PLS platform and is showing really great data. So, it's really flexible. The only thing we haven't done at this time is like anti-inflammatory drugs for like rheumatoid arthritis, but I think that would be a good one, obviously. We haven't delivered antigen in a vaccine. We've only delivered adjuvant, but obviously that could be done as well. And for the same reasons, it might be a really good idea. So yeah, I mean, that's, when you think about it, that you're targeting inflammation because you're targeting the immune system here. The sky's the limit, because you think of so many diseases that have an underlying inflammatory basis. And just off the top of my head, atherosclerosis. So the next question was about what diseases we can image, right?
Yes.
Right. So just off the top of my head, atherosclerosis has a major inflammatory component, immune cell accumulation. There's no doubt in my mind that this wouldn't be a great system to image atherosclerotic plaques, though we haven't done so yet. Rheumatoid arthritis could be imaged. Cancers can be imaged. We already have some data on that. And that's, again, our thinking that, boy, this probably should go to tumor-associated macrophage. And indeed it does. We don't know whether it's going through the lymphatics to the tumor or directly into the tumor. That's another, set of hypotheses we're looking at. But this really, it's, I often, I said this in the past when I was talking about therapeutic applications of this technology. I've been in nanotechnology, nanomedicine for 15 years. I've, through the NCL, looked at over 350 different concepts, or particles anyway, and a lot of them were purported to be targeted, okay, to a tissue. Very rarely did we see evidence of targeting, and in cases in which we did, there were often problems with drug release or other, issues with successful therapy. Nanoparticles in general are very good at targeting inflammation in the lymphatic system, especially if you use receptors such as this that are really geared towards getting at, we spent so much time trying to stay out of the lymphatic system, right? I think once you look at it the other way and you're trying to better optimize achieving lymphatic distribution, it really has opened up a whole new area of research for me, and the lab. It's really been interesting.
Terrific. Could you also target sites of infection?
So absolutely, this will go to sites of infection. Yeah, we already have evidence of that. Yeah.
Okay, terrific, awesome. So 2 more questions. I know we're getting close to the top of the hour, but I definitely want to make sure all the questions get answered. So if anyone else has any more questions, just please put them in. We'll stay on to make sure we answer everything. Are there any issues that might complicate development or regulatory approval?
So the only concern right now is that we're dealing with a new chemistry, right? This is brand new. There have been succinylated proteins in the past that have been administered clinically, but there's never been anything like a linear, polyamino acid construct like this. So even though we don't see any safety concerns from the studies that we've run, you never know, of course, until you get closer to primates or closer to man. That's a concern, the lack of clinical experience. But besides that, we're dealing with a highly biodegradable polyamino acid backbone structure. This structure can clear very, very rapidly. Okay, so in the case of the 10K construct, where you just want it to go to lymph node and then leave the body, it does so almost immediately. So there's no risk of bioaccumulation. Scale up should not be an issue because of the simplicity of the chemistry and the high yields of click chemistry, which has been used, classically for scale up, and is sought after by medicinal chemists for the purpose of scale up because it's simple chemistry. This is shelf life stable. So if you lyophilize this material, we've never had an issue with any conjugate we've made in which you can keep it in the fridge or the freezer, and we've seen no evidence of degradation. So that's a real big hurdle that we don't seem to have. So you put that all in perspective, and I don't really see any showstoppers. We don't use any, all types of solvents in the chemistry are easily removed, during the lyophilization process. This can be sterilized prior to lyophilization, so we really don't have issues. It can undergo filter sterilization. It's a stable construct. So yeah, I don't see any concerns.
Okay, awesome. So you've hinted at the clearance, but how is the polymer cleared from the body?
Well, we know the fact that we're seeing the larger polymer constructs, being the 62 and 25K, starting to go into the urine at early time points, not as rapidly, as efficiently as the 10K, which makes sense. We know that these larger polymers must be degraded because they're too large to undergo that glomerular filtration. So this suggests that clearance really isn't an issue because these apparently are metabolized quickly enough that they're eliminated within a couple hours, but still manage to do their job, still manage to get to where they should go. I'm talking about systemic clearance, not, again, as I mentioned, not the lymph node clearance. Lymph node clearance is not something that we've evaluated yet. I would surmise that lymph node clearance would undergo the same process that we're seeing systemically. So that would involve proteases and peptidases and cleavage to smaller units that can then be cleared. These are very polar molecules, and so we anticipate that as they are chewed up, become smaller and smaller, polar small molecules are generally cleared renally. We see that for the 10K. We see that in some fashion systemically for the larger constructs. I assume that's what's going to happen in tissue too, though we haven't specifically looked at it at this time.
Okay, thank you. So we'll move to the last question then, and I'll just remind everyone, if you could fill out the survey that I've sent out, that would be awesome. You can click on it now and fill it out after the session if that works best for you. So maybe you could just use this, Dr. Stern, as your summary too of everything you see that people should remember. But I'll ask you this question, and you can cover that in your answer as well. So what are the greatest advantages of this platform over others you have seen in your 15-year career in nanoscience?
Well, Okay, so first I would say the rapid targeting. Okay, so that's very unusual. Out of 400 projects, I can count on one hand the number of times I've seen effective targeting. Okay, so the targeting occurs rapidly, whether it's sub-Q, intradermal, or IV. Okay, that's also unusual. I don't know that I've ever seen that, that type of route, non-dependent, route of administration. Also, in cases where I've seen targeting, very often the constructs are very complicated, the chemistry is very complicated, and it's very difficult to characterize. These linear polymers are extraordinarily easy to characterize. And, we are the nanotech characterization laboratory. We spend a lot of time characterizing materials, right? This is some of the simplest material that we've ever characterized. And we are very good at characterizing it orthogonally by several different, methods and coming to the same answers, which show that this is the chemistry is very rugged. The yields and loading support the fact that it's a very simple chemistry, but it's very easy. The drug loading is much higher than I've seen in the past. And I guess I would wrap up with saying that, I always go into a project looking for liabilities, right? Because I've worked with so many different projects and everything's got its issues. And trying to identify issues, at this point, I would really say it's just the lack of clinical experience, because it just seems to overcome all the other ones that I've seen coming along, especially for targeted constructs, but even for non-targeted constructs. So that's where I would end it. I think it's a very promising technology. Certainly, I think it should be developed because, I've seen so many different concepts over the years, and this one just really stands out. And I would, of course I'm biased, I would say, because it's, it's a technology we developed in-house, but yeah, specifically to try to get rid of all the liabilities that there could potentially be based on what we've seen in the past. So that's how we narrowed down to, we're going to do a prodrug, a polymer system. We're going to rely on targeting that involves the vasculature. We really checked all the boxes getting to this point. So it wasn't serendipitous. I wouldn't say that things are working well. It was a long process of trying to, really optimize what an ideal targeted platform would look like. So I'll just end with that.
Great, awesome. I was going to say in a more layman's way, but fundamentally, when someone partners with you or goes through a licensing opportunity with you and your team, where they're not just getting the science and the technology, but your hundreds and hundreds of, very focused projects and your smarts to basically say when they kind of try to step over the line in some area, you go, no, we've tried that before and it's just not going to work. So
exactly. And that's what we do. That's our bread and butter. What we do on a daily basis is help bring other people's products to market. And in the process, we gain all this knowledge over the years of what the issues are, what are the stumbling blocks, how to get around them, and that's what we do for a living. And certainly, we would apply that knowledge to any licensing partner, especially with a platform like this that we're very familiar with. Would be easy to do.
Yeah, and I'll also refer to the fact that you're calling it a platform, which we've discussed is the third opportunity. It's a therapeutic, it's a diagnostic, but also has the opportunity for someone to take this on as a platform technology to implement all the different therapeutic options and all the different diagnostic options. So to me, it could be quite a game changer, but that's just my personal opinion.
Well, thank you.
Great. Well, thank you again, Dr. Stern. Thank you again to Joe Conrad and Lauren for their participation in helping bring this forward. And again, you have the phone numbers and the email addresses of everyone if you want to make any connections, or if you want me to connect you directly, you can also reach us at 1 Million Solutions in Health. So again, thank you so much, Dr. Stern and everyone. And I wish you a great rest of your week and much success as we move forward with this.
I thank everyone for their interest, and thanks to everyone at NIH for providing this format for inventors. It really is helpful.
Awesome. Have a great week, everyone, and we'll see you at the next event. Take care. Bye.